It's visible, Dr. Rajaram Garu.
Yes, sir. Good afternoon, one and all. Thanks to Skill AP and our principal sir and our training and placement in charge, Vishwanath sir, for giving me this opportunity. Myself, Dr. Rajaram Mohan Rao, who is working as associate professor in the Department of Pharmacology in Vigyan Pharmacy College. So today's topic is biomedical research. So what is meant by biomedical research? Here, biomedical and research. So in this topic, we'll be going to see what is meant by biomedical, because there are three kinds of research among biomedical research. One is basic, next is applied, and third one is clinical. So what is meant by basic? So there are n number of processes that are being done in our body from time to time, taking respiration or means metabolism and so and so. So it is study of process of life. Study of process of life is nothing but what are the things that is being going on in our body at any time, like after taking food, what is being secreted. So evaluation of all these, checking all these comes, and the prevention and treatment of diseases. So what are the possible things to prevent a disease? What are the treatment options for treating a disease, and what are the genetic and lifestyle changes that is happening, impacting our normal body health. So types of research is of three types. One is basic, applied, and clinical. So basic is nothing but what is there in the things. What is exactly means how we came to know mitochondria is the center for respiration, where ATP is produced, that is being evaluated. How we know ribosomes are the sites where protein is getting synthesized. So these are the things to check what is there in the life cells is known as basic. So what is meant by basic life sciences? What is there exactly? What is happening in normal stages? Like where our main things is genome. So where genes, translation, transcription, protein synthesis, like lysosomes are what they are destroying the means if there is any pathogens, they are the removing cells. Identifying all these things, what is actually going in our body, this is known as basic research. So research conducted to increase fundamental knowledge. Fundamental means what is there, not directed towards solving any particular problem. His main thing is we are not going to check any problem. We are evaluating what is there, like respiration in mitochondria, protein synthesis in ribosomes, genes are present. These are the directing things, what to synthesize, what not to synthesize, and immune factors, what are there, what are their functions. These all the things coming under basic research. So focuses on understanding normal life processes and diseases. This may be molecular or cellular. These main things. Many of the times there are people who are being acknowledged with Nobel Prizes because of their basic research, because they are the people who is bringing what is there in the cells. They are the people who is working vigorously. So this may be cellular or molecular. So this provides building blocks for other types of research is the first stage. So many of the like DNA. So applied is nothing but take one example of diabetes. So what is meant by diabetes? There are two kinds of diabetes, type 1 and type 2 diabetes. In type 1 diabetes, our pancreatic beta cells have been destroyed because of autoimmunity, whereas type 2 because of insulin resistance. So this is the things where means scientists have identified that insulin plays a crucial role in glucose uptake by the cells. This is basic research. They have identified that because of autoimmune disorders, type 1 diabetes have been came. This is basic research. Based on availability of basic research, we will be directing some medicines or some diagnosis or some devices. That is known as applied research. What is existing we are identifying means basic research based on basic research, because people have come to know that because of insulin is a hormone which is helpful in uptake of glucose by the cells. So if there is insulin abnormality or decreased insulin levels, chances of type 2 diabetes or type 1 diabetes. So we are identifying based on this information, we are synthesizing some molecules or taking some plant compounds. In type 1 diabetes, only insulin. So we'll be giving insulin analogs, whereas type 2, we are, because of insulin will be there, but because insulin resistance. So we'll be synthesizing medicines or identifying many plant extracts and giving, so it will stimulate or increase the and power of insulin. So this is applied research. Applied is means based on the basic knowledge, we are directing towards some specific objectives, directed towards specific objectives such as development of a new drug, treatment, or surgical procedures. So applied research is being conducted because based on the basic information, it will be in animals mainly in first, because before going to humans, because of safety concerns and ethical issues, first we'll be doing on animals, tissue cultures, computer models, and the last humans. So clinical is the last phase after having some basic information that we have obtained from computers based, in vitro based, in vivo based. After getting sufficient data of the particular molecular devices, after it being safe, evaluated as safe, we will be going for clinical phases. So usually the last stage of medical research, clinical is the last stage of medical research. So it is used when other forms of research have been taken place, means before going to the human trials, we will be conducting initially in computer software in silico simulator, next in vitro, in vivo, at the end human trials. So it is useful for treating potential drugs and treatment in humans. So it is on what it is based, because of the build up based on the basic and applied stages, because it builds on what is done in basic and applied stages. We will be going for clinical phases. So it may be taken place in a hospital or a clinical setting for healthcare, because as it is being in clinical trials, we'll be testing new molecule or new devices. As it is new, we don't know what may be the toxic effects we will be getting in that. So sorry for the interference. So clinical, where clinical will be doing has been performed in hospital or clinical setting or healthcare, because as in clinical trials, we will be checking on the new molecules or new devices for which we don't know exactly how it is going to behave, how it is going to affect. So in controlled supervision of health professionals, so in the hospitals where all the health professionals will be there, they will be evaluating the new drug or new devices. Applies directly to, means what kind of things it will be, maybe for prevention, means prevention comes like vaccines, because when we are checking for vaccines, it is for prevention many diseases, where diagnosis like devices I have said, medical devices. So based on the medical devices, we will be diagnosing various kinds of diseases. Now we will be everyone be knowing about what I can say, insulin glucometers, or so such kind of things, or treatment of a specific disease in individual or group of individuals, or means maybe preventing like vaccines, or diagnosis like blood glucometer, or treatments like various medicines which we are being using for various kinds of disease conditions. So broad varieties of activities and areas are being checked in the clinical levels. It is subdivided into 3 types: human clinical trials, psychosocial and behavioral research, disease control research. So human clinical trials, here we will be evaluating for a new moiety or new drug or medical devices, whereas psychosocial and behavioral research, this kind of research that we will be seeing a number of articles in our general newspapers. They will be checking their behavioral parameters based on the heat, temperature, based on the mingling with people, based on the stress activities, based on the emotional things. That is psychosocial behavioral research. Here there is no medicines involved, but they are checking the behavioral parameters based on the environment they are exposed, based on the environment, based on the stress conditions, work, emotions, relations. Disease control research, it is like nothing but a particular disease. So if you are taking such kind of things, if you are having a very good diet, so what are the chances of getting a specific disease? If you are having low fat diet, what is the chances of getting hyperlipidemic? And second group, they will be evaluating like hyperlipidemic. So they will evaluate if the people are being consumed with high protein diet, good fiber diet with less fat diet, whether the people are getting hyperlipidemia or the people who are taking more fat. So they will be developing like disease control research study for a particular disease, like human clinical trials for specific device or medicines, psychosocial behavioral research, where they will be evaluating for social behaviors, and disease control research. So clinical trials in a nutshell. Before going for a clinical trial, we have to get approval for the what are the protocols we are going to follow during the performing clinical trial. So investigator selection, after investigation selection, we will be getting approval process. Next, after approval process, based on the protocol, we will recruit the patients, and we will be dividing based on the number of patients. We will allocate based on the groups and study groups, and after giving our drug of choice or devices, drug treatments, we will enter the data and review the things, and after that we will calculate the data into a using statistical analysis. Based on that, we will calculate that whether it is statistically significant or not, because there are two kinds of instances: a person in disease without drug, a person with disease with drug, but the drug has to show tremendous activity, not a slight decrease. That time only the medicines will be accepted, only if it is showing significant activity. After getting significant results, if the molecules which are showing good results, they will be screened for next stages. If it is not having very good activity, it won't be screened for next stages. Next, presentation and publication, and after based on the data, we'll be submitting to the final form to the FDA, where our drug approval process will be in hands. So what are the methods that are we using in biomedical research methods? One is chemical, mathematical, or computer simulations, in vitro tests, non-human animal models, human studies, and epidemiological studies. So what is meant by chemical, mathematical, and computer simulations? Coming to the preclinical trials, initially before starting clinical trials, initially we will be checking the molecules which is in the data library, because as a chemical scientist used to secrete n number of chemical compounds, and you might be knowing that drug receptor interactions. When a drug binds to the receptor, then only we will be having the activity. That may be, so drug must have the drug must have affinity and intrinsic activity. So we'll be having a number of chemical compounds. So we'll be checking, means based on the basic research, we will be coming to know what kind of disturbances in been happen. So what are the signaling pathways is there? Signaling pathways is nothing but, if you are coming for what I can say G protein coupled receptors, there are a number of pathways. If it is G stimulatory, it will be activating adenyl cyclase. After adenyl cyclase, ATP will be converted to ADP. After this, phospholipase, protein kinase. These are the things in the downstream pathways. So we will be targeting one molecule. So we will check whether our drug is going to hit that specific compound or not, specific receptor or not. So first we will check what is the structure of receptor and what is the structure of our molecule, so that only it can fit. Now, if it is not fitting, if it is not binding, there is no activity. So as we are having a number of compounds, and we also be knowing the structure of the receptor, we will do in the chemical formula. It means computer simulation. So we will check, we will draw the diagrams of the chemical moieties, and we also know the structure of the receptors. We will check whether it is fitting or not, So we will first synthesize n number of compounds. After synthesizing n number of compounds, based on the NMR, mass, chromatographies, we will be identifying the structure. After getting the structure, we will do docking studies. Docking studies is nothing but we will do in the computers. We will, where the receptor structure will be available and our chemical structure also will be available, based on that how many number of fitting, so whether it's fitting properly or not, and based on that we'll be going mathematical calculations. Next, after completion of this, okay, which molecules fit into the specific receptors, then we will go for in vitro tests. In vitro test is nothing but outside the living cells or glass. So in vitro test is nothing but the experiments that are being on glass slides or petri plate dishes. Next one is non-human animal models. Next one is after performing in what else I can say, glass or petri plates. Next we will be going for animal models. Next, human studies. So it is proven most useful in the preliminary research stages, where scientists can stimulate ideas about new research directions, because as first we have to synthesize molecules, because the structure of receptor will be unique, so it will be fixed. So based on that we will synthesize n number of compounds, so whether it is fitting or not, which is fitting perfectly based on the chemical moieties. We will be knowing if it is this moiety is present, we will be having toxicity. If it is this moiety is there, because receptor also having n number of functional groups, our drugs also will be having n number of functional groups, if it is docking properly or not, binding is proper or not. So it proves very helpful to stimulate new ideas about new research directions, helpful in understanding complicated biological principles and dynamics, because after binding only it will be having effect. So dynamics means what drug does to the body. So provide ideas about new research directions to pursue, So analytical chemistry tests detect a substance or measure its potency. So initially we will be synthesizing chemical moieties in the laboratory. After synthesizing, we will be performing what I can say chromatography. So based on the chromatography, we will be separating n number of compounds. So after isolating each individual compound, we will be identifying the structure of the compound. After identifying the structure of compound, same docking. So useful in developing vaccines, prescription drugs, and vitamins. So what mathematical and computer models? So can predict biological response on the basis of chemical structures and activity in an organism, because we will be knowing the structure in the computer simulator and our chemical structure and the receptor structure. Based on that, whether it is fitting or not, both structures are similar or not, how much attachment it is there, we can predict. So based on that, we can estimate how biological effect can be. If it is fitting properly, lock and key model, if it is getting perfectly fitting, so we can estimate, okay, it will be very good. It can have very good effect. So the biological effects of chemicals can be quantified and correlated with the chemicals, biochemical properties, composition, and structure. So based on these properties, we can calculate how much efficacy can be based on using mathematical models. So what are the strengths? Because you might be knowing, to get a drug approval or to complete process of drug testing, it takes 14 years to 16 years. So computers can increase the speed and efficiency with which data is used and processed. Because you might be knowing, recently we are almost the entire world is affected with COVID pandemic. So many people are at the back of vaccines, but not at the back of medicines, because vaccines is nothing but we are just remodifying or manipulating the virus itself. In COVID, many people are feeling that Anandaya medicine is a great boon, but it is very good. But you might be have to know the difference between what is vaccine and what is drug. So vaccine is nothing but we will be taking the virus in basic kinds. So what is the major thing in COVID? Because as virus enters into the body, as time progresses, the numbers will be doubled, tripled in exponential manner. As it is new to the body, we don't know what kind of antibodies or what kind of immune system we have to activate so that we can eliminate the virus. So in vaccines, what we'll be doing, we'll be taking the viruses and we'll make them inactive. So whatever the quantity we are giving, that will be the final number. As time is getting progress, the number of viral particles will be increased in the active conditions, whereas in what I can say, in vaccination conditions, the number will be final. So it will be easy for our body to identify. You might be knowing memory cells will be processing the virus particles. It will have a part of it, and whenever virus enters into our body, our memory cells will be having the particles, so that our body can synthesize what kind of antibodies, what kind of bullets we have to synthesize, so that we can eradicate virus. So with vaccines, we will be with vaccines, this is not sure that we won't get disease. We will be getting disease, but the severity will be not that much severe. Because what is that? Because as virus enters into our body, it will multiply, but our body is in such a position that it can eliminate. Coming to the drugs, so why only all over the country people are focusing on vaccines? Why many people are not focusing on drugs? Why focusing on vaccines? Because vaccines were having ready-made. So take virus, inactivate it, and give it to the patients, so that in an inactive form, it won't be multiplicating. So our immune system, we have sufficient time to synthesize the bullets to kill the virus. Coming to the medicine, that is not the procedure, because we don't know which kind of drug will have impact. As we are having surrounded by n number of plants, n number of chemicals, we don't know which may suit, which can inhibit. So as it is complex process, as we are having flooded with n number of chemicals, so initially to reduce the number, initially we'll go for chemical computer simulation. So in the computer simulations, we'll be screening millions of compounds. So the compounds which are fitting to the receptor only, we will be taking the things. Where if it is same thing now, for COVID vaccine, means COVID medicines, if we want to identify the medicine also, it takes a number of time and lot of time, because we have to check each medicine in the, means if it is animals, means we have to induce COVID disease condition in the animals, we have to check each and every medicine. So it takes a lot of time. So the scientists have identified that if we dock in the computers, so what are the spike proteins, so what are the receptors in the virus, so and based on the what are the chemicals, what are the chemical moieties we are present, so they will be docking. So what are the things fitting, so we will be identifying. As a result of this, lot of burden on the companies will be reduced and time also saved. Next, after screening the things in the computer simulator, we will be identifying which are getting fitted. Next, we will go for, so computers increase the speed and efficiency with which data is used and processed, because we can screen n number of compounds in a short span of time. Pattern recognition programs enable scientists to compare characteristics of one compound to another, because as we are knowing the structures and we also know the spike protein chemical structures, so we can be identify which pattern which suits to the better, So pattern recognition programs enable scientists to compare characteristics of one compound to the another, which compound is better, which compound is not effective. That is the reason now, as we are not having any medicines for COVID, we are just doing repurposing of the existing drugs. Repurposing is nothing but what are the drugs that are existing in the market, we are reusing the things. So they are checking. Sometimes they used to say initially antibiotics are very good, ivermectin is very good, it is things, but as the research is going progress, is saying based on the, they have checked initially there is no time, so they used to suggest all the compounds, all the antibiotics. As the time getting progress, they are having some time to evaluate whether it is actually having efficacy or not. Next, they can extrapolate data, because based on the things they can identify, okay, as it is getting fitted, so it can be having very better efficacy. So from high dose experimental exposure to low dose, and from animals to humans, reduce the number of animals needed for research, because as millions of plants is there to check which plant is. So you might be knowing, if anyone is having cough, what kind of compound we used to use? We used to add turmeric. We are not taking other plant now, because some of our ancestors have screened n number of plants, they have tasted each and everything. I finally identified, okay, turmeric can have some effect. So as in the computer simulation, it will reduce from lakhs to thousands. So if it is reducing from lakhs to thousands, what will happen? So that we can reduce the number of animals. Now if it is more compounds, we have to take in more animals. As number of compounds are reduced, we can reduce the number of animals which is required for preclinical research. So what is the limitations of computer based? Cannot replace laboratory testing. Just it is based on chemical structure, we are thinking that okay, both can fit, but in the actual real life, it will be different, because here there is no surrounding environment. There is no surrounding environment. Based on the environment, our mood will be fluctuating. If it is hot environment, we will be very irritating. If it is a cool breeze, we will be getting very sleepy. As in the both situations, we are the same people, but based on the cool environment, we are unable to wake up. Whereas hot environment, we feel very irritating. So that is differs now. So in a computer simulator, only the structures only, but environment will be missing there. Next, computers do not generate data. They only process the existing data, because they are not generating the data. They are just processing, because they are processing matlab means we are giving the structure of the drug and we are know the structure of receptor. So they are just a processing whether binding or not, but they are not generating new kind. They are not making any new things. They are doing only what we have given, that is giving the things. What we have given input, whether fitting or not, that only. Computers equipment and software is expensive. So these are the limitations, because it cannot replace laboratory testing. Laboratory testing is the final things, and it cannot generate data because it can generate only process the existing data, and computers, equipment and software is expensive. So next one, in vitro testing. In vitro is nothing but in glass. So takes place in an artificial environment. So in vitro things, we will be separating any organ or cells. So we will be culture. Many people might be knowing about tissue culture. Tissue culture is nothing but we will be taking any tissues, dividing means as tissue contains a number of cells, we will break the bonds between tissues so that we will get individual cells, and we will be culturing the cells. As we're culturing, the cells number will be increasing continuously, and we'll be checking if it is cancers. What is the property of cancer cells? It used to increase the number based on the time progress. If I have inoculated initially 10,000 cells, and I will be giving the nutritious environment so that cells won't die and they will be getting nutrition. As time progresses, what is the property of cancer cells? It have multiplication potential. So, and I will be checking my drug of interest. So whether the number of cells being decreased or not, or it is maintaining same, or whether it is increasing. Based on that, we'll be doing the in vitro tests. So the term used for any biological processes or reaction that takes place in artificial environment. It is artificial environment. It is not natural. Natural means existing body things. So that is natural. We are replicating. We are making a living things in an unnatural way. We are making unnatural things in an unnatural way. So the term used for any biological processes or reaction that takes place in an artificial environment. So the cells or tissues being studied originally come from a living organism. I have taken, we have said no cancers. So if you want to check any breast cancer, we will be separating the breast from the patient who are having breast cancer. We used to lyse the tissue into cells and we'll be culturing the things. Cell cultures can be, we sometimes will be cells or sometimes tissues. Cell cultures, isolated tissues and organs are used in early and intermediate stages of biomedical research. Sir, why we are doing this in the early stages? Because we are not playing with the lives of any animals. See, as what is the property of cell culture? As we are giving nutrients, whatever the things we are taking in the body, we are giving the things. So the number will be continuously increasing. So n number of studies we can perform without sacrificing any animal, because there are also lives. That is fact. So this will be helpful in reducing the number of usage of animals. Potential remedies are first tested in this manner to discover how they interact with cells and tissues. Potential remedies like that I have said now. In the computer simulated, whatever the compounds that have been attached very nicely, we should check this because they are the potential remedies. So as fitted in the chemical means computer, whether it will have same efficiency in tissue culture or cell culture or not. So what are the strengths and limitations of the in vitro studies? It is able to identify single effect of a substance in isolation. So it is able to study single effect of a substance in isolation, because effect of single substance means if you are taking any drug, many people might be knowing through oral route, what happens, there is a topic called first pass metabolism, because as we have taken medicine, it goes into the through oral cavity, esophagus, stomach, intestine, from intestine, liver, from there into portal venous system to the heart, from there to all the circulation. So before going to the systemic circulation, some part of the drug will be metabolized in the liver, some part will be in the stomach, some part will be intestine, majority will be in liver. So whatever the amount of drug we are giving, it will be metabolized. There are chances of chemical modification also. There is chance of chemical modification because n number of compounds. Stomach will be having acidic pH, bicarbonates in the intestines. They may have, because acids and bases, they may react with the chemical moieties. Chemical moieties, they also will be having a number of chemical compounds. So as we are having this number of chemical variations in our body, there are chances of interacting. Whereas in cell culture, that kind of effect won't be there. Only cells and the things that is required for both of the cells and the chemicals which we are testing the things. So such kind of things won't be there. So able to study single effect. There is no other factors that is impacting the drugs that we are checking. Less expensive, less time consuming, more accurate and more readily controlled than in vivo. In vivo is nothing but whole animal. So it is less expensive, less time consuming, because at a time We can screen n number of, if you're taking one petri plate, if we take 1,000 petri plates, I can check in 1,000 compounds at a time. Whereas in the animals, it is not necessary, it is not sufficient. We can't check within 1 day or 2 days. For every study, it takes minimum 1 week to n number of weeks. Whereas in cell culture, within some 2 hours, 48 hours, we can get the results. Less time, because for any corporate world, time matters a lot compared to money. Time matters a lot. So less expensive, less time consuming, more accurate and readily controlled. Readily controlled means, see, if they have taken any new moiety, if there is having any toxicity, it will be a lot of effect. If it is whole animal. So such kind of things is not there. We can control the things. We can control the things like, see, if we have taken any drug through oral, if it is not having, if it is having some side effect, we can get through vomiting. Or if we are given through injections, what we can do? Nothing. Now we can't control in whole animals. Whereas in vitro, if the things are going worse, we can take into the control. Able to control temperature, because we'll be having the controlled conditions. Whereas living things, we can't control the temperature, because based on the things, based on the external environment, see, you can check the things. If the outside environment is cool, our body used to have warm temperature. If the outside environment is warm, our body used to have warm temperature. So such kind of temperature variations, that won't be there. You'll be having uniform temperatures. Able to control temperatures, we can maintain acidity, oxygen levels, and environmental conditions of cultured cells. So cell cultures often contain cells that can replicate themselves in the laboratory. So we will be doing in the studies only where the cells can replicate. So that only we can identify whether if it is replicate in itself, it is indicating that it is in live condition. If it is in not live condition, they won't be replicating. But replication potential will differ from cell to cell type. Cell type to cell type, replication potential will be different. But we'll be doing only when the cells are in multiplication, because multiplication indicates the cells are in living conditions. The cells are not in dead conditions, because we used to remove all the initial, when the cells we are adding, we used to give growth media. As growth media used to time to consume, and metabolites also used to come to the cell culture media. So we used to remove the cell culture contents, because as nutrients will be exhausted, we used to change regularly the nutrients. We used to supply the nutrients. Critical to the study of viruses, because viruses can grow only in living cells. We can't check viral studies in vivo animals. We can't control, because if you are doing any animal research, we are studying any virus. If suddenly it sneezes, it is not in our hand. Animals can sneeze, and viruses are the most deadly combinations which affects many people's life. So whereas into the cell culture, it will be in controlled conditions only, so it will be easy for checking those things. So critical to the study of viruses, because viruses can go only in living cells. Limitations. What are the limitations of in vitro studies? Biochemical processes leading from chemical exposure to toxic effects cannot be duplicated in vitro, because what the things is, if we are having headache, how we used to take, majority of the times we used to take what I can say Amrutanjan or Jandu bomb, but many people used to take Saldon, that is to go through orally, from there to stomach, intestine, hepatic, blah blah blah, throughout the entire body. But that, as we are taking medicines, but it is only touching the head, it should not disturb any of the part of the body. So in vitro means we will be checking on the only specific cell, like if it is headache, what are the blood vessels is there, which is relaxing, because headache is because of vasodilation. So we'll be checking on the vasodilation properties. So in the cell culture, we're checking on only on that. Whereas in complete animal, as we are giving drug, it is to go to pass through the entire body. So we can't identify, other than the target cells, we can't check the things. If we are having headache, it may damage, what I can say, many people used to take paracetamol for what I can say for fever, antibiotic. Sometimes they used to take for what I can say headache also. But liver means paracetamol causes severe hepatotoxicity. Liver can means paracetamol can cause severe hepatotoxicity. So we can't check such kind of things in the in vitro cultures, because we are not checking for liver in there. We are checking only targeted cell or organ. Cells grown in culture are not exposed to other functions taking place in living organism, because they are not exposed to any kinds of metabolism, any kinds of chemical changes. Cells do not metabolize toxins in cell culture in the same way they do in the whole body. As we are taking our medicines, they used to get eliminated after some point of time, so that the concentrations will be reducing time period. So that only we used to take, if for some medicines we used to take thrice a day, some medicines twice a day, some medicines once a day. Whereas in vitro, such kind of metabolism is not there. So the chances of toxicity, difficult to maintain differentiated cells in a culture, because differentiated cells is nothing but from same cell we used to get different kinds of means like stem cells. Stem cells from same cell we used to get n number of cells, because you might be knowing if there is any, what I can say, any kind of threat, robbery, anything, if the person's hair or nail or blood stain is sufficient enough to identify, because any part of the body contains same genome. Any part of the contains same genome. Based on the organ, specific genes will be expressed. Don't be confused. What I'm saying is, during a threat or the theft or any robbery, if the police find any hair or nail or any blood stain, anything, they can identify the, what I can say, they can identify now who is the culprit. See, 3 different samples, but how they identified? Because all the cells contain same genome. All the cells contain 23 pairs of chromosomes, same genome. But in different organs, different set of genes will be expressed. That is differentiating. That is differentiating. So cell culture can't differentiate into different things, and cell cultures cannot generate sufficiently reliable data about how a substance affects complex interactive system. Because as the same thing, if it is going through oral, n number of complex systems, it has to transverse, n number of organs it has to displace. So such kind of complex systems won't be there in vitro studies. But it gives idea, basic idea. So we have to check this one also, but basic our motto is whether it is curing the things or not. If it is cancer, if it is preventing the cellular replication or not, cell division or not. That idea we will get initially. That idea we'll be getting from the in vitro studies. Next, non-human animal models. Next one is non-human animal models. Animals provide the best known surrogate for humans in the laboratory. These are the replicates which will mimic humans. Why? Because our genome and their genome is around 95 to 98% similarity. 95 to 98% similarity. That's why we are checking. Chimpanzee is somewhat more relevant. That's why now we are checking on the animals. Vital research model, because they provide the whole complex living system that can interact and react to the stimuli such as to the humans do. As we are reacting to n number of compounds, this is also having the capacity to interact. Similarities between animals and humans outweigh the differences. The similarities are major in number compared to the differences. So that is why we are going for animal studies. That is and which animals is showing, we are checking on the same, we are evaluating on the specific animals, not on the what are the animals that is being available in the planet. So strengths of animal models, because to do work on humans, there are a lot of n number of ethical procedures before performing a clinical trials. Many times we used to check so-and-so company have conducted a clinical trials, but they have not taken care of health of the general public. So such kind of ethical issues will be there. But animals, to get a approval to perform experiments on animals also, we must have ethical approval, but less stringent. Ethical alternative to using humans. Animals provide a whole integrated complex biological system. In vitro, it won't provide whole system. Whatever the organ systems we are having, animals also having digestive system, reproductive system, respiratory system. So whatever the systems we are having, this also used to show. Animals provide a whole integrated complex biological systems. Animals share the same structures. Whatever the tissues we are having, whatever the cells we are having, it also same in animals. Scientists can design experiments where they can control for more variables than with human. So based on this data, based on the similarities and somewhat less stringent rules and availability numbers, so scientists can plan work so that for identifying new drugs or medical devices. So clinical studies is nothing but where we are performing on human volunteers or human subjects. So most often used in developing prescription drugs. So at the end stages only, we will be going. Earlier, we are not going to check into the humans. Most often used in developing prescription drugs. Looking for the answer. So is the drug biologically active in humans or is the drug safe in humans? So what are the disease means before taking into the, what I can say, clinical trials, our motto is it has to be safe and it has to be efficacy. It has to be safe and it has to be efficacy. But initial, what we need is it has to be safe compared to efficacy. Because if it is having toxic and it is having efficacy, so if it is more toxic, less efficacy, they won't be considering because it is of no use. But more benefits, less side effects, it will be benefited. So we'll be checking in humans what are the side effects, what are the how the effective the drug that we have means. And one more thing, in computer models, lakhs and millions of drugs will be screening. From there it will be coming few thousands. In vitro studies where cell cultures, from there to hundreds to tens, 100 compounds, from there to animals 50 to, it will be coming around 50 to 10 to 50. From in humans 10, from 10 to it will be coming finally 2 to 3 compounds, 2 to 3 moieties. Among 2 to 3 moieties, finally they will be selecting which is better. So what they'll be checking is the drug biologically active in humans and is the drug is safe in humans or not. So what are the different phases of clinical trials? So there are 4 phases of clinical trials: phase 1, phase 2, phase 3 and phase 4. So there is one common question that in which means clinical trials is nothing but we are checking the drug effects on human volunteers, human subjects. So in the first phase, we are checking on which kind, means there are 2 kinds now, diseased and healthy. So in which kind of human volunteers we are going to check? Which kind? Suppose if you want to evaluate anti-diabetic drugs. So whether you want to be checking on diabetic patients or healthy patients. Social like bar, whether it is in diabetic or healthy patients. So we'll be in first phase, clinical phase, first one, we'll be checking on healthy volunteers because first our motto is, see, as I have said, for headache will be as it is having in the head, we used to take through oral route. As the drug transverses n number of pathways systems and finally reaches head, and it will adjust the things that has been altered in the head. That will adjust what the things have been altered in the head. So means as in clinical trials, we are checking on the new drugs. We don't know what are the side effects, what are the toxic effects. So if you are giving for a healthy person, after giving to the healthy person, we should check after giving the drug after 24 hours or 48 hours or 70 hours, we should check the parameters based on the blood. We should collect blood from them. We should check whether cytokine levels are in high condition or not, whether immune system is active or not, whether BP has been differentiated or not, whether sugar levels are what happened. So such kind of things will be checking, because as the healthy drug has to be, means if drug is safe, it should not disturb any of the organ systems. It has to go, it has to do its work. It should not touch or disturb any of the major organ or any of the other activity. So if it is safe, it won't disturb any. So if it is healthy, we can identify. If there is any variations in the blood parameters, we can identify that, okay, drug is not safe. It is causing some abnormalities. Whereas if we check directly in the diseased patient, we don't know, because most of the times we'll be having comorbid condition. The people are having diabetes, they may be having some serious related complications. They will be having what I can say kidney related complications, eye related complications, brain related complications. We don't know because of which happened. Because of drug, because of disease, we don't know. Because a number of complications is to come as the time progresses. So if it is what I can say, if it is healthy patient, if the drug is safe, it will go inside, it will come outside without disturbing anything. As it is, he's not having any disease, it's just going and coming. So because first our main motto is how much safety it is. So in the phase 1 clinical test, what we'll be doing, determine the drugs interactions with the human system. Determines the drugs interaction with the human systems. involves a small number of healthy volunteers. As it is in normal conditions, we don't need to evaluate in n number of people. N number of people. It takes around 1 year. It takes around 1 year. Number of people is 10 to 20 in the phase 1 clinical trials. Phase 1 clinical trials. Next one is phase 2 clinical trials. Phase 2 clinical trials. So here, as in initial one, safety will be identifying. Here we will be checking for efficacy studies and dose determination studies, whether it is efficacy or not. And we also identify what dose we have to give, because metformin is 500 mg, glipizide is 5 mg, paracetamol 650, 500. It differs now. So what dose we have to give for the humans to treat? What dose has to be given for pregnant women, children, and pediatric old patients? We have to identify. So controlled tests that help determines the drug effectiveness on certain organs. So phase 2, drug effectiveness, whether drug is effective or not, and dose, what dose we have to give for humans. The number of people here is involved is 10 to 100 to 300 volunteer patients. Here, as we are checking for efficacy, efficacy means disease condition has to be there. Then only we can check for efficacy. There is no disease condition in phase 1. We are not checking any efficacy because there is no disease condition. So what it will do? It is not touching any organ, not any system. So simultaneous to animals and human tests to assess safety. It takes around 4 years. Phase 2 clinical trials takes around 4 years. Phase 3 clinical studies. Phase 3 clinical studies, it also involves safety and efficacy studies, majorly efficacy studies and safety studies. It will be done in multicentric, means it will be done in n number of countries, because as I have said, now based on the climate conditions, people used to express different kinds of genes. In some people, the metabolism will be slow. Some people, metabolism will be high. So based on the climatic conditions, so we used to check. We used to do in multicentric, different parts of the world. So phase 3 done to confirm the results of earlier tests and identify any adverse reactions. Because phase 2 also efficacy. So we for confirming how much efficacy is there. Clinical for this, as it is multicentric, number of people involved here is more in number, around 1,000 to 3,000 volunteer patients. It takes around 3 years. After this, based on the summarizing data that we have got, we should apply for the new drug approval, for new drug application to the FDA. It takes around 2.5 years to complete. So it takes around 2.5 years to controls. So what are the strengths of human drugs? Have actual human data, because as our drugs are being, means for whom we are developing for testing on humans. So we are checking on the final. But what are the limitations is ethical. It is not such easy to test any new drugs because lot number of ethical issues. Numerous variables which may affect test data are introduced when humans are used, because we might be not knowing means what happens when we keep any steel or stainless steel things in the rain or any water where water is there for a long time. Initially, if you're keeping for 1 week to 10 days, we are not seeing any rust. As the time progresses, if it is good quality, no rust. If it is less quality, chances of rust. So in animal trials, we used to monitor every time what kind of food they are getting, what kind of temperature they are there, what kind of oxygen levels. Whereas in human subjects, after completion of trials, as I have said, we can't see rust within 10 days or 6 months. It may take years. So in humans, we can't keep the humans in isolated or segregated positions for longer times. So n number of numerous variables which may affect test data are introduced when humans are used. So next, next topic is handling of laboratory animals and routes of drug administration. So as we are pharmacology in pharmacy level, so we'll be concerned about preclinical trials. So here we'll be checking about how the rules of drug administration on animals, how to handle the animals, how to make them very user friendly. So as the animals are very means having more mobility, so it is very hard to handle it because it having the mobility, so it escapes easily and very likely to bite, because you might be knowing rats are very sharp. Rats and mice are having very strong teeth. It can bite very easily. It can bite very easily. So how to catch? Because our intention is to check or to evaluate the drug in the animals. So to give our drug, we have to hold the animals. We have to hold the animals. We can't give with the water bottles where we used to give for children. Children, we won't giving tablets. No? We used to dissolve in that, and we used to keep in water bottles. If we keep in water bottles, because of medicines, the taste will be different. It won't be pleasant. So animals won't drink water. So we have to enter in, means we have to give the drugs forcefully into the animals. So how to handle the animal to give you a drug? So grab at the base of a tail. Check here. So grab at the base of a tail. Allow 4 feet to grasp the edge of cage or table, because if animal is holding any item, it will be very easy, because if it is holding means it is won't be moving forward. If it is not holding, we won't be getting any grip, because if it is not holding means it will be continuously moving position. So allow 4 feet to grasp the edge of cage or table. So that one we used to keep all the time. So upper tray of the, what I can say, animal cages, so that animal it can hold the cage, so that we can hold the animal very easily. If it is not holding, it will be moving continuously. Continuously, it is very difficult to catch the things. So how to approach this? So approach from behind and place forefinger and index finger on either side of neck. Why we have to keep on either side of neck? Because if it turns head, we can, our fingers will be very close to the means we should not let the mice to stretch to rotate its neck. If it is rotating its neck, it will catch our fingers. It will catch our fingers. Such a position we have to hold that it won't move its, what I can say, its head. So approach from behind and place forefinger and index finger on either side of the neck. So we'll be holding on both sides. We'll be holding on both sides. So this side pressure and this side pressure. So both will be in strong position. So the animal, it won't be moving head right side or left side. We'll be holding like this. So animal will be in this position only. We'll be holding here. Here we'll be holding here. So it is very hard. It is very hard to catch. That means to catch our fingers. And we have to hold, but we should not be very forcefully. Because of fear, some people hold like very things, so that it muscles and means bones will be breaking. So approach from behind and place forefinger and index finger on either side of neck, exerting pressure on the mandibles. Exerting pressure on the mandibles. Hold rest of the animal against palm with remaining fingers. So after holding rest of the animals with this palm. So here animal will be holding, animal, rest of the animal. Here head will be, head, both side of the neck will be holding here. Rest of the animal will be here. So rest of the animal will be in here. So see. So like this. Presentation capture. Now I shared the presentation. Whether check it once again, whether presentation is visible now. Okay. Thank you for the info. Wait a minute, is presentation is not being visible. Now it's visible. అది కాదు వేరేది అవట్లేదా వెయిట్ ఏ మినిట్ ఓకే వెయిట్ ఏ మినిట్ ఓకే ఓకే So how to hold the animal? So how to hold the animal? Grab at the base of the tail with one hand and allow 4 feet to grasp the edge of cage or table. Allow 4 feet to grasp edge of the cage or table, because if it is not holding the cage or any table, the animal will be in moving position. If it is in moving position, we can't hold it. We can't get it right. So if it is holding, we will get some grip. To get grip, so approach from behind and place forefinger and index finger on either side of the neck, because we will be having means what I can say. If we are holding here, I means from this side and this side. So my head won't be moving, because if it is holding this side, I can't move this side. If it is holding, so head will be in this position. It won't be changing its direction, because if it is changing means it can catch our fingers. It can catch our fingers. So we'll be holding on both sides of the neck. So approach from behind and place forefinger and index finger on either side of the neck, exerting pressure on body of the mandible. Exerting some pressure means to hold some pressure. So here our what I can say, neck will be there and rest of the body will be in here. Rest of the body will be here, present in here. So hold rest of the animal against palm with remaining fingers. So like this. Next, that is one approach. This is another approach. It is full restraint. Whereas here, only here will be holding. Just will be touching the animal in this manner. Touching in this manner. Here holding. Whereas here, complete skin will be holding. Complete skin it will be holding. Whereas here, this two will be working. It is just touching the things. Just touching the things. See, just you are holding here. We are holding here. Whereas here, we will be taking whole skin into our fingers. Whole skin into our fingers. There only we are stopping the head to rotate. Here, whole body it will be in our position. Body also can't move. Body also can't move. It is known as scruffing technique. It's a restraint, full restraint. So hold by tail base. Use forefinger and thumb to gently pin down the head. Use the same fingers, grasp a sufficient amount of loose skin over the neck, and hold tail with little finger and finger of the same hand with additional support. So we'll be holding here, rest of things here, and taking the tail as it is mice within our hands, it will be there. So I will with the tail, I will be holding this. So tail also it won't move, because tail is there means it used to rotate. It used to make this kind of movements. So here head, rest of the body, tail I used to rotate here. So the complete body is in our hands. So next one is collection of blood. So what are the different methods? Because why collection of blood? Because to identify the parameters, because if it is taking an example of blood glucose, we have to measure. So we have to collect blood. There are a number of disease conditions where inflammation plays a crucial role. So whether inflammatory mediators are high in number or decreased in number because of drug medication or not. To identify lipids, mineral amounts, hormonal levels, we used to take what I can say blood and we used to estimate. So for collecting blood, there are different methods from where we can. So from humans, they used to collect from veins. From animals, different areas. They used to collect blood from tail vein, from orbital sinus, from eye. Blood collection from cardiac puncture, because heart is the organ which used to pump blood to the whole body. Heart will be having sufficient blood, so they used to puncture heart, and they used, because in the blood, heart में blood होता है ना, so in the heart blood will be there, so we used to puncture and we used to take the blood. Blood collection from saphenous vein is present in the thighs, intraperitoneal injections, subcutaneous injection, oral feeding, and sexing. So blood, these are the top 4 is blood collection. Rest of the things are injections, because injections is nothing but giving out drugs for evaluation. Giving our drugs for evaluation, intraperitoneal injection, subcutaneous, oral feeding, or so blood connection, blood collecting from tail vein, tail. So in tail, so the vein will be very, so for means mice or rat tail is very small, so the veins also will be very narrow, very narrow. So the blood we will be getting is very minute amount, because vessels it's a very small, so that the amount also will be getting is very minimal in number. So for collection of small amount of blood, approximately 0.1 ml, 0.1 ml. So before collecting blood, we used what are the things we used to do? Because animals used to be like a social animals, it will be having in the same cage 5 to 6 mice. There only their litter will be there, everything will be there. So whole body will be in somewhat what I can say filled with many of the dust and foreign particles and unhygienic conditions. So if we cut the tail, so we will be making injury, and if we are making injury, so it will be contaminated. So the tail also will be, means the cutting part will be exposed to n number of microorganisms and chances of infection. So before that, we used to sanitize the thing. So the sanitizing is nothing but we used to remove all the infection, we used to remove all the pathogens. So for that, 75% alcohol we used to use, so that we, by dipping cotton, we used to wipe the tail, we used to wipe the tail, so that all the pathogens will be dead. If the pathogens are there, if we cut after cutting the tail will be exposed, we will be cutting some injury. So chances of pathogen entry and chances of harm to the animals. Because as we are following ethical, means what I can say, as we have to treat animals like our friends, we have to maintain some ethical principles. So 70% alcohol cotton ball for surface disinfection, and small plastic bottle with 0.5 centimeter diameter holes in both ends as mouth restrainer. Next slide, I will be showing what mouth restrainer. Next is scissors for cutting the tail, and pipette for collecting the blood, and vial for blood collection. So we will be holding like this the mouse as we are collecting blood from the tail. So as I have said, restrainer means animals will be strained, closed, tightly packed. So see here, as I have said, it is of 1 centimeter to 2 centimeter diameter. So we will be allowing what I can say mice into that, and we will be having here what I can say 4 to 5 holes, 4 to 5 holes, 4 to 5 holes. So that, because as we are keeping in restrainer, as with this having tightly packed, we should not make them to suffocate. So that only here you will be having n number of holes, small holes, so that it will get sufficient oxygen. And here we will be closing, and here we will be closing. Here we will be closing. So to the lid, we will make one hole. It is like a bottle, and it is a cap. To the cap, we will make one hole, so that we can take the tail out. We can take the tail out. Here we will be making a number of holes to get animals without any suffocation. Leave the tail of the mouse outside and cover the strainer. So cut the tip of the mouse tail by scissors. Tip the mouse tail by incisors, and blood will be coming. So with the help of pipette, we can collect the blood. We can collect the tip. We can collect the blood with the help of the pipette. Next one is blood collection from orbital sinus in mouse. Orbital sinus in mouse. So it is one of the easiest. Compared to tail, it is very easy and less harmful to the animal. Why, sir? Why it is less harmful? As we are cutting tail, and we are means here, see, you will be seeing that we are collecting from the eyes. Sir, if something small kind of minute particle, if that falls, we will be having a lot of pain. If it is means, see, we are having some injury, and you are having small dust particle on the eyes, which will make you more scary. A small particle in the eyes make you more scary. Whereas in tails, it is not like that. Now, so here, what is there? Now, here, retroorbital sinus will be there. Sinus vein will be there. So we'll be just making a like injection, like we are piercing sinus vein, and we'll be dragging the blood. After that, we will be remove the collecting tube, so that the vein will be closed naturally. Whereas in tail, we are cutting the tail. We are cutting the tail. Whereas here, we are just keeping needle with the vein, collecting the blood, removing the needle. The vein will be closed. No issues to the eyes or no issues to the veins. Whereas in tail, we are cutting the tail. We are cutting the tail. a convenient and easy method for blood collection in mouse. As it is veins, we will be getting a lot of sufficient blood, like 0.5 ml, 0.5 ml. So before that also, we used to wipe the eyes, close the, we used to close the eyes, we used to wipe with cotton cloth, and we used to make all the things sanitized. So 75% alcohol cotton for surface disinfection, 27 gauze needle with 1 ml syringe for injection, and glass capillary tube and vial for blood collection. See, for blood collecting, from what I can say, from retroorbital, it can be done with anesthesia or without anesthesia. We can be doing with anesthesia or without anesthesia based on your expertise. So if you are scary about the things, you will go with anesthesia. To make less scary, we can go without anesthesia. But to get blood without anesthesia is far better model. So anesthesia mouse by intraperitoneal injection. So giving anesthetic by intraperitoneal injection after giving sufficient time. So the animal will be in, what I can say, somewhat consciousness it will be losing. After that, open the eyes and PA means keep capillary tube. Many people might be knowing capillary tube. Capillary tube is nothing but in one side, if you keep any, if capillary tube is placed in any liquid, based on capillary reaction, the liquid will come outside of the other, what I can say, other side of the tube. So if we are placing any capillary tube, any liquid in one side, the other side of the side of the capillary tube, liquid will be coming out. Here, capillary tube will be kept into the, what I can say, into the retroorbital. So one side of the capillary tube will be in the retroorbital, retro means sinus vein, and the other part will be kept under the, what I can say, blood collecting tube. So through the capillary action, blood will be flowing to the capillary tube to the other part of the. So use a sharp end glass capillary tube to penetrate the orbital conjunctiva and rupture orbital sinus. So see here, so one side will be kept inside of the eyes, and the other side, as it is because of capillary reaction, blood will be coming out, so that we can collect blood with vial, So see how they are collecting. If we are following proper procedures, retroorbital, we will get sufficient blood without causing any damage to the eye. Make it sure that without any damage, not minimal damage, without any damage to the eye, we can collect blood n number of times. Blood collection from cardiac puncture in mouse. Blood collection, as heart contains sufficient blood. This procedures means, what I can say, in retroorbital, after collecting blood, we can leave the animal. It can be living condition. Whereas if we are collecting blood from, what I can say, from the heart, we have to collect only from the heart in the last stages, before sacrificing of the animals only. At that situations, we used to collect blood from the heart. So because we are collecting blood from the heart, we will be getting a lot of volume compared to the tail and retroorbital. We will get around 1 ml of blood within a short period of time. Must be performed under general anesthesia, because as we are keeping our needle into the heart, it must for anesthesia. It is a must to keep the animal in anesthesia, because as we are piercing means animal can't tolerate. As it is in eyes, animal can tolerate. There also pain will be there. If it is in cardiac puncture, animal can't tolerate the pain. So we have to keep it in general anesthetic condition. So we used to clean the area by using alcohol, needle. So 27 mg needle with 1 ml syringe for injection, and 24 gauze needle with 3 ml syringe for blood catheter. What is syringe for injection? Syringe for blood catheter. Syringe for injection is nothing but we are giving anesthetic agent for injection, and 24G needle for blood catheter. As blood have property of blood clotting, as the higher number indicates narrow diameter. Higher number indicates narrow diameter. So injection needle, anesthetic injection needle diameter is less compared to blood catheter needle, because blood has the property of coagulating. So if its diameter is bigger, so less chances of coagulation. So anesthesia mouse by intraperitoneal injection, and After, as you might be knowing, heart will be in continuously beating position, although in anesthesia. So after giving anesthesia, so animal will be losing what I can say sensations. We will place artificial respiration. You can check here artificial respirations. You can check here artificial respiration, and we will be opening the. We will keep both hands, means both palms apart, and we will check where the palpitations are coming, where heart is beating. We can check, because how people can know heart is here or left side or right side based on the heartbeat only. So we will be checking with our fingers that where heart is beating more, where heart is beating more. After identifying the spot, we used to insert the syringe and we used to collect blood. So search for the maximum heart palpitation with your fingers. So check where the heart is beating more. Means a heart is having 1 centimeter, around 1 to 1.5 centimeter. So to collect blood from appropriate area, we will check with our hands. So where it is getting sufficient beat, we will insert the needle, we will collect the blood. So here, inserting 24G needle through the thoracic wall at the point of maximum heart palpitations. So withdraw blood slowly by your right hand. So other with our right hand. So blood collection from saphenous vein in mouth. So saphenous vein is nothing but it will be in our thighs. So this method is used of multiple samples are taken in the course of day. As if you want n number of samples, we used to follow this procedure, and also we used to follow what I can say retroorbital method also if it is having n number of blood collection samples. Because in some conditions, if it is checking for metabolism studies, how drug is getting metabolized. So we will be knowing now. So 2 hours 20%, 4 hours 60%, 8 hours. Based on that only now we'll be taking 1 drug per day or 3 medicines a day. So at that time we have to collect n number of bloods so that we can identify how much blood is there, how much drug is there in the blood. So saphenous vein blood collection helps in multiple time points. So same things, what I can say, we used to clean it with the sanitizer so that any less chances of cross contaminations. Placing a nose on cage lid and grasping loose skin behind the ears with your thumb and forefinger. So we'll be keeping animal in a restrainer, where, as I have said, it is in thigh. So as it is in thigh, as animal is having hair, we used to remove the hair around the thighs. Pull out the leg and remove the hair by a trimmers. So hair can also be removed by shaving or trimmer or shaving by using a small scalpel. So the saphenous vein is seen on the surface of thigh. So the saphenous vein is seen in the surface of thigh. So after seeing this, we can collect n number of times blood. So apply Vaseline after disinfecting the surface area to reduce clotting and coagulation during blood collection. So 24G needle is used to collect blood. 24G needle because more diameter. So approximately around 100 microliters of blood can be collected each time, and n number of times we can collect the blood. So up to this blood collection, now giving drugs. Which are the areas we should give drugs? So the most common route is, one is intraperitoneal, other is oral or peroral. So it is the most common method of administering drugs to rodents. Peritoneum is nothing but below diaphragm. We will be having peritoneal cavity, lot of air space. Why we are giving to the peritoneum means we can give large amounts of dose, and in peritoneum there are no many vital organs. As there is only empty space, we are not damaging any organ. So same. So place a mouse on cage lid and grasping the loose skin behind the ears with your thumb and forefinger, and we'll be checking here. So here in this area, we'll be giving here up to here only all the vital organs will be available, whereas here only empty space, only peritoneal cavity. As soon as mouse head is restrained, the mouse can be picked up and tail secured within your finger, because we also used to hold tail also, because as we are tail is also in free condition, it used to make movements. So if it is movements, it is very difficult for us to insert. It is difficult for needle to be inserted to give the doses. So we used to give the injection sites should be in the lower left quadrant of the abdomen, because Vital organs are absent from this area. Only tip of the needle should penetrate the abdominal wall to prevent injection into the intestines. So we can give maximum dose of 1 to 2 ml. Next one is subcutaneous. Subcutaneous is nothing but in humans. Many people used to take subcutaneous route. What I can say, insulins. So for subcutaneous, we will be giving only when we are checking for what immunological studies. Here we can't give means more volume of drug. That is only skin layer, as it is very thin animal. We can't give large volumes compared to peritoneal cavity. So see, we'll be taking the mouse. We'll be giving subcutaneous just under the skin. We'll be giving. So whether drug has been given or not, how we will be identifying is, as it is only giving in the peripheral layers, we are giving some volume. So we will get some knob. As it is peripheral layers only, we are giving some liquid. So that area will be bulged. A lump of injection substance can be seen through the skin after injection. A lump of injection substance. So this is another method. If it is in dorsal side like this, if it is ventral side, means see here. Peripherally, we will be giving subcutaneous injection can be made to the scruff of the skin. Next one is oral feeding in mouse. Oral feeding in mouse. So this is one of the major things. Means most of the times people used to follow either oral or intraperitoneal. For means for parenteral route intraperitoneal, for top means for oral route. So gastric intubation ensures that all the material was administered. So feeding amount can be limited to 1% of the body weight. If it is 20 grams, we can give around 1 ml or 2 ml of the volume. So for this, we should have gloves. So we should give like this. We have to make sure that head is in straight position, is not in bending position. If it is in straight position, chances of going needle into the thorax, not into the esophagus. If it is by mistakenly going to the esophagus, thorax, you won't be knowing now. So what it is, so if it is going to the lungs, food is going to the lungs, chances of this. So if it is head in straight position, the closure will be perfect, so that drug will be directly going to the esophagus, not into the thorax. So some complications. See here, we will be checking. So the person unable to keep, what I can say, the needle went into the thorax, not into the esophagus. That's why animal will die in the spot. If by mistakenly, if you have given to the oral instead of esophagus, if you are given to the thorax, because see, a person can survive. If it is heart failure also, they can survive for some days, some months. But if person can't take respiration, can they survive for a minute? They can't survive. No? So by mistakenly, if you are given into the thoracic cavity, thoracic means to the lungs, it can't exchange air, it can't exchange gases, the animal will die spontaneously. So be very cautious while giving about drugs to the oral cavity. So how we differentiate between male or females? See here, genitals. So the distance between genital orifices and anal root is greater in males compared to females, whereas in females, means genitals and orifice, the distance is very less compared to the males. So if it is more distance between the genital organs and anal root, means anal, so that it can be male. If it is less space, it is females. So see here, it is anal, it is genitals, it is male, whereas here the distance is very small. So thank you. Thank you one and all for giving me this opportunity to deliver the topics. If you have any doubt, ready for the answers.
Good afternoon, sir. Hello, Raman sir. Good afternoon, sir. Am I audible? Hello. Hello.
If anyone have doubts, I will share my mail ID in the comment section, or if you can ask about more issues.
Hello.
Hello.
So good afternoon, sir.
So good afternoon, sir.
Ramahan, sir, am I audible?
Yes, sir.
Sir, I'm Ramatra from ABSSTC, sir.
Okay, sir.
Sir, Google form posters, sir. Fill చేయమని చెప్పండి, sir. It is considered as a attendance.
Okay.
Chat box లో పెట్టండి, sir.
Okay, okay. Dear students, fill the Google form for feedback so that you will be getting the attendance. As Google Forms is being shared in the comment section, guys, just go to the comment section and fill the Google form.
Students, do you have any questions? You can ask, please.
So I kept my mail ID in the comment section. If you can have any doubts, you can ask me or you can mail me if you have any doubts. So someone is asking to resend the, what I can say, Google Forms.
Yes, sir. Already it is posted thrice, sir. I'll post it again. Just give me a moment.
But thanks a lot for Skill AP, AP SSGC, and Vignan Pharmacy College for giving me this opportunity. And thanks to
Thank you,
our
sir.
Vishwanath sir.
సార్ మీరు లీవ్ చేయదు సార్, మీరు హోస్ట్గా ఉన్నారు. మీరు లీవ్ చేస్తే సెషన్ ఎండ్ అయిపోతుంది.
ఓకే సార్.
అంటే ప్రతాప్ సార్ మీద మిమ్మల్ని హోస్ట్ చేశారు. అంటే వీళ్ళు గూగుల్ ఫామ్ ఫిల్ చేసేస్తే జస్ట్ 5 minutes సార్, వీ కెన్ క్లోజ్ ద సెషన్.
ఓకే సార్. అంటే నేను ఇప్పుడు ఏం చేయమంటారు సార్? గూగుల్ దాన్ని ఏం చేయమంటారా సెషన్ని?
మీరు చేస్తే మొత్తం ఎండ్ అయిపోతుంది సార్.
అది నేను చేయలేను సార్.
ఒక 5 minutes నుంచి.
రాజారాం గారు, హలో రాజారాం సార్.
సార్ చెప్పండి సార్.
What I request you is you can talk 1 or 2 minutes about tomorrow's just basic introduction, what you are going to teach and everything.
Okay sir. Okay sir.
So that the participants will have an idea.
Okay sir. Okay sir. Definitely sir. So about tomorrow, today we have what are the different kinds of researches and what are the different kinds of routes of administration. So tomorrow session we are going to deal what kind of animals that are going to be used in preclinical trials, on what basis, because as a n number of animals is there, on what basis we should select which kind of animals for which kind of studies, what are the pros and cons of each animal species. And we are having on Pharma software which is given by PCI, which used to be, so we'll be using that software and we should be demonstrating some of the experiments that used to be come in our B.Pharm syllabus. So these are the two topics. One is different kinds of animal models where we used to conduct preclinical trials, and the software where we are doing some of the experiments.
Thank you, sir. Thank you.
Okay, sir.
Google form.